Hey everybody, I am going to be talking about section 10, which is spontaneous generation and biogenesis. Just as a disclaimer, if I look over here a lot, it's because I have a script so that this video doesn't run 10 minutes long. So we are studying spontaneous generation and biogenesis. Originally, the idea our question we are answering is where cells came from. Originally, the idea that organisms just spontaneously generated was the theory that was believed. This was dated back to 328 BC with some of Aristotle's writing, where they believed and based on just their observations that things just spontaneously generated. So it wasn't until 1626 when Francesco Redi questioned this theory that things spontaneously generated by using an experiment using flies, that he questioned if spontaneous generation is actually true. So in his experiment, he used various different flasks with meat because it was to be believed back then that flies spontaneously generated from meat. So he questioned that and used the closed systems in various flask containers to see that organisms are created from other organisms. So in this experiment, you see that the flies laid their eggs became maggots, and those maggots became other flies. So this experiment sparked that organisms come from other organisms. It wasn't until the 1800s with most notably Charles Darwin, that we started to question how those organisms came from other organisms. Darwin studied the theory of evolution, or the process of organisms adapting and changing to their environment over a set time that eventually formed new types of organisms. In his study, he went to the Galapagos Islands and studied different type of tortoise and finches. And in his study of finches, we were able to see that all of these finches share one common ancestor, but because of what they ate, it made them into distinct, different species. So some of these finches are insectivores, meaning they only eat insects, while some eat large seeds and some eat small seeds. And because of that difference, they created different species of finches. From studying characteristics of different organisms changing over time, two sets of scientists challenged and tried to recreate how life on Earth could have formed gradually from non-living chemicals. This is specifically the Oparian Hall-Day hypothesis, where he hypothesized that the early sea was like a primordial soup that because of the different living chemicals in the ocean, different non-living chemicals combined together to create earlier life. It wasn't until Miller-Urey actually challenged this experiment and created a closed system where they created the ocean and atmosphere of the early Earth to create those what we know now as proteins. So they created a closed system, used water for the early sea and evaporation, sparks for some type of electric current, and mixed a mixture of gases, including ammonium, hydrogen, and methane, to create amino acids, which are the building blocks of proteins, ultimately cellular life. So from this experiment in the 1950s, we go to a scientist named Lynn Margulis in the late 1960s, who questioned how individual cells could eventually evolve from this. When studying cell theory in the symbiotic theory of cells, which is the theory that a cell, an animal cell, evolved from a large anaerobic cell and engulfed a small aerobic bacterium, creating what we know as the mitochondria, and that plant cells evolved from an anaerobic eukaryotic cell that engulfed a photosynthetic bacteria, creating chloroplast. She studied these two organelles specifically because they are respectively the powerhouse of each individual type of cell. Now, this is just an overview of section 10. I would go back into your MAAPS book and read each individual section so that you can better understand how these theories build off of one another, and how these scientists were able to come to their own individual theories.